Yeah, hello friends, Unit 9 Biotechnology. Chapter 9 Biotechnology Principles and Processes. Chapter 10 Biotechnology and its Applications. Ever since the days of Rene Descartes, the French philosopher, mathematician, and biologist of 17th century, all human knowledge, especially natural sciences, were directed to develop technologies which add to the creature comfort of human lives as also value of human life. The whole approach to understanding natural phenomena become anthropocentric. Physics and chemistry gave rise to engineering technologies and industries which are all worked for human comfort and welfare. The major utility of the biological world is as a source of food biotechnology. The 20th century offshoot of modern biotechnology, the 20th century offshoot modern biology changed our daily life as its products brought qualitative improvement in health and food production. The basic principle underlying biotechnological processes and some applications are highlighted and discussed in this unit. Herbert Boyer was born in 1936 and brought up in the corner of western Pennsylvania, where railroads and mines were the destiny of most young men. He completed graduate work at the University of Petersburg in 1963, followed by the 3 years of postgraduate studies eight at Yale. In 1966, Boyer took over assistant professorship at the University of California at Francisco. By 1969, he performed studies on a couple of restriction enzymes of the Escherichia coli bacterium with especially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends on the strands. These clipped ends made pasting together pieces of DNA practice precise exercise. This discovery in turn led to a rich and rewarding the conversation in Howley with the Stanford scientist named Stanley Cohen. Cohen had been studying small ringlets of DNA called plasmids, and which float about freely in the cytoplasm of certain bacterial cells and replicate independently from the coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and then rinsed setting them in other cells. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in desired configurations and insert the DNA in a bacterial cells, which could then act as manufacturing plants of for specific proteins. This breakthrough was the basis upon which the discipline of biotechnology was founded. 9 नंबर का हमारा चैप्टर बायोटेक्नोलॉजी प्रिंसिपल्स एंड प्रोसेसेस. 9.1 प्रिंसिपल्स ऑफ बायोटेक्नोलॉजी. 9.2 टूल्स ऑफ रिकॉम्बिनेंट डीएनए टेक्नोलॉजी. 9.3 प्रोसेसेस ऑफ रिकॉम्बिनेंट डीएनए टेक्नोलॉजी. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful of humans. In this sense, making curd, bread, and or wine, which are all microbes mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense. Genetically modified organisms to achieve the same on a larger scale. Further, many other processes techniques are also included under biotechnology, for example, in vitro fertilization leading to the test tube baby, synthesizing of gene and using it, developing a DNA vaccine or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology EFB has given one definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organisms, cells, parts of thereof, and molecular analogs for products and services. 9.1 principles of biotechnology. Among many the two core techniques that enabled birth of modern biotechnology are: first, genetic engineering techniques or to alter the chemistry and genetic material, DNA and RNA, now to introduce this into host organisms and thus change the phenotype of the host organism. Second, by process engineering, maintenance of sterile microbial contamination, now free ambience of in chemical engineering processes are to enable growth of only the desired micro eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appropriate the advantages of sexual reproduction over sexual reproduction. The former provides opportunities for variations and formulation of unique combinations of genetic setup, some of which may be may be beneficial to the organism as well as the population. Sexual reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organisms. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in a progeny cells of the organism, but when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the aligned piece of DNA has become part of chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of any aligned piece of DNA in an organism, it needs to be a part of chromosomes, which has a specific sequence known as origin of replication. Thus, an aligned DNA is linked with the origin of replication so that this aligned piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning and making multiple identical peoples of any template DNA. Let us now focus on the first instant of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant the DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a negative plasmid or autonomously replicating circular extra chromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors. restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA act as vectors to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into human body. In the same way, the plasmid can be used as vector to deliver an alien piece of DNA into the host organism. Now, the linking of antibiotic resistance gene of DNA into the host or the alien linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which acts on cut DNA molecules and joins their ends. And this makes a new combination of circular autonomously replicating DNA created into in vitro of and is known as recombinant DNA. When this DNA is transferred into Escherichia coli, the bacteria closely related to salmonella. It could relate using a new host or DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene is Escherichia coli was called cloning of antibiotic resistant gene in Escherichia coli. You can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable genes, introduction of identified DNA into the host, maintenance of introduced DNA in the host, and transfer of DNA to its progeny. 9.2 Tools of recombinant DNA technology. No, now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is restriction enzymes, polymerase enzymes, ligases, vectors, and host organism. Let us try to understand some of this in detail. 9.2 Restriction enzymes. In the year 1963, the two enzymes are responsible for restricting the growth of bacteriophage. In Escherichia coli were isolated. One of these added methyl groups of the two DNA, while the other cut DNA. The latter was called restriction endonucleases. The first is the restriction endonuclease in the two whose functioning depended on a specific DNA nucleotide sequence was isolated and characterized. 5 years later, it was found that in the two always cut DNA molecules and at a particular point by recognizing a specific sequence of six base pairs. This specific base sequence is known as the recognition sequence of Hindu. Besides Hindu, today we know more than 900 restrictions enzymes that have been isolated from over 230 strands of bacteria, each of which recognize different recognition sequences. The convention of for naming these enzymes is the first letter of the name comes from the genus, and the second two letters come from the species of the prokaryotic cell from which they were isolated. Example: Icoara comes from Escherichia coli R113, and in Icoara the letter R is derived from the name of strain. Roman numbers following the names indicates the order in which the enzymes were isolated from that strain of bacteria. Restriction enzymes belong to a larger class of enzymes called nucleases. These are of two kinds: exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make haircuts or at specific positions within the DNA. Each restriction endonuclease functions by inspecting the length of the DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Figure 9.1. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. Do you know what palindromes are? These are groups of letters that form the same words when read both forward and backward. Example, Malayalam as against a word palindrome where the same word is read in both directions. The palindrome in our DNA is a sequence of base pairs that reads us the same on the two strands when our orientation of reading is kept the same. For example, the following sequences reads the same on the two strands in 5 prime to 3 prime direction. This is also true if read in the 3 prime to 5 prime direction. Restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands. Now, this leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends of on on each strand. Figure 9.1. These are named so because they form the hydrogen bonds with their complementary cut counterparts. Now this stickiness of the ends facilitates the reaction of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources genome. When cut by the same restriction enzymes, the resultant DNA fragments have the same kind of sticky ends, and this can be joined together end to end using DNA ligases. Figure 9.2. You may have realized that normally, unless one cuts the vector and the source DNA, which are with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under the an electric field through electric field through a medium matrix. Nowadays, the most commonly used matrix is agarose, which is the natural polymer extracted from seaweeds. The DNA fragments separative the resolve according to their size through sieving effect provides by the agar gel. Hence the smaller the fragment size and the further it moves. Look at the figure 9.3 and guess at which end of the gel the sample was loaded. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light, and without staining you can see bright orange colored bands of DNA in exposed to UV light. Figure 9.3. The separated bands of DNA are cut out from the gel piece. This step is known as elution. The DNA fragments purified in the way are used in constructing recombinant DNA by joining them with cloning vectors. In cloning vectors, you know that plasmids and bacteriophages have the ability to replicate within bacterial cells independent of the control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. Some plasmids may have only one or two copies per cell, whereas others may have 15 to 100 copies per cell. Their numbers can go even higher if we are able to link to an align piece of DNA with bacteriophage or plasmid or DNA. We can multiply its numbers equal to the copy number of the plasmid or bacteriophage. Vectors used at present the are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. The following are the features that are required to facilitate cloning into a vector. Origin of replication ORI. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cells. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover the many copies of the target DNA, it should be cloned in a vector whose origin support higher copy number selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced in a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc. are considered useful selectable markers of Escherichia coli. The normal Escherichia coli cells do not carry resistance against any of these antibiotics. Cloning sites in order to link the align DNA, the vector needs to have a very few preferably single recognition sites. For the commonly used restriction enzymes, presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. Figure 9.4. The ligation of aligned DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at a BAMH one site of a tetracycline resistance gene in the vector PBR322. The recombinant plasmid will lose the tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformers on the tetracycline containing medium. The transformers growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow and in ampicillin containing medium, but not on that containing a tetracycline. But non-recombinant will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistance as gene gets inactivated due to insertion of aligned DNA and helps in selection of recombinants due to inactivation of antibiotics is nice cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability now to produce a color in the presence of chromogenic substrate. In this, the recombinant DNA is inserted within the coding sequence of an enzyme beta galactosidase. This results into inactivation of the gene for synthesis of this enzyme, which is referred to as insertional inactivation. The presence of a chromogenic substrate gives blue colored colonies. If the plasmid in the bacteria does not have an insert, presence of insert results into insertional inactivation of the beta galactosidase gene, and the colonies do not produce any color. Now, these are identified as recombinant colonies for vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lessons of transferring genes into plants and animals from bacteria and viruses which have known this foreign genes and how do deliver genes to transform eukaryotic cells and force them to do what the bacteria or the viruses want. For example, agrobacterium tumefaciens, the pathogen of several record plants is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct this tumor cells to produce the chemical required by the pathogen. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. Better understanding of the art of delivering genes by pathogens in their eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. The tumor inducing TI plasmid of agrobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to the plants, but is still able to use the mechanisms to deliver genes or interest into the variety of plants. Similarly, retroviruses have also been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into suitable vector, it is transferred into a bacterial plant or animal host where it multiplies. 9.2.3 competent host for transformation with recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cells must you first be made competent to take up DNA. This is done by treating them with a specific concentration of a divalent cation such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. Recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius, heat through shock, and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. This is not the only way to introduce alien DNA into host cells. In a method known as macro injection, the recombinant DNA is directly injected into the nucleus of an animal cell. In another method suitable for plants, cells are bombarded with high velocity micro particles of gold or tungsten coated with DNA in a method known as biolistics or gene gun. And the last method uses disarmed pathogen vectors which are when allowed to infect the cell transfer the recombinant DNA into the host. Now that we have learned about the tools for constructing recombinant DNA, let us discuss the process facilitating recombinant DNA technology. 9.3 Processes of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonucleases, isolation of a desired DNA fragment, ligation of the DNA fragment into a vector, transferring the recombinant DNA into the host, culturing the host cells in a medium at large scale, and extraction of the desired product. Let us examine each of these steps in some details. 9.3.1 Isolation of genetic material DNA. Recall that nucleic acid is the genetic material of all organisms without exception, and majority of organisms is this is deoxyribonucleic acid and DNA. In order to cut the DNA with restriction enzymes, it needs to be in pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as the RNA, proteins, and polysaccharides, and also lipids. This can be achieved by treating the bacteria, cells, plant, and animal tissue with enzymes such as lysozyme, bacteria, cellulose, plant cells, chitinase, fungus. You know that genes are located on long molecules of the DNA intertwined with proteins such as histones. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatments and purified DNA ultimately precipitates out after the addition of chilled ethanol. Now, this can be seen as collection of fine threads in the suspension figure 9.5. 9.3.2 cutting of DNA at specific locations. Restriction enzymes digestions are performed by incubating purified DNA molecules with the restriction enzyme at the optimal conditions for the specific enzyme. Agar gel electrophoresis is employed and to check the progression of restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves towards the positive electrode. I note figure 9.3. The process is repeated with the vector DNA. Also, the joining of DNA involves several processes after having cut the source DNA as well as the vector DNA with a specific restriction enzyme. The cut out gene of interest from the source of DNA and cut vectors with the space are mixed and ligase is added. This results in the preparation of recombinant DNA. 9.3.3 Amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vitro using of two sets of primers, small chemically synthesized oligonucleotides and that are complementary to the regions of DNA and the enzymes DNA of polymerase. The enzyme extends the primers using the nucleotides provides in the rest reaction of the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. That is 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from the bacterium Thermus aquaticus, which remain active during the higher temperature, induce denaturation of double-stranded DNA. The amplified fragment, if desired, can now be used to ligate with the vector for further cloning. Figure 9.6. 9.3.4 Insertion of recombinant DNA into the host cell organism. 9.3.5 Obtaining the foreign gene product. 9.3.6 Downstream processing. Then summary. Biotechnology principles and processes. Summary. Biotechnology deals with large scale production of marketing of products and processes using live organisms, cells and enzymes. Modern biotechnology using a genetically modified organisms was made possible only when a man learned how to alter the chemistry of DNA. Made possible only when a man learned how to alter the chemistry of DNA and construct recombinant DNA. Now, this key process is called recombinant DNA technology or genetic engineering. This process involves the use of restriction endonucleases, DNA ligase, appropriate plasmid or viral vectors to isolate and ferry the foreign DNA into host organisms, expression of the foreign gene, purification of the gene product, that is the functional protein, and finally making a suitable formulation for marketing. Large scale production involves use of bioreactors. बायोटेक्नोलॉजी प्रिंसिपल्स एंड प्रोसेसेस 9 नंबर का हमारा चैप्टर है जिसमें हमने पहले से देखा प्रिंसिपल्स ऑफ बायोटेक्नोलॉजी, रूट्स ऑफ रिकॉम्बिनेंट डीएनए टेक्नोलॉजी, क्लोनिंग वेक्टर्स, origin of replication, selectable marker of cloning sites, vectors for cloning genes in plants and animals, and the competent host for transformation with recombinant DNA, processes of recombinant DNA technology, isolation of genetic material DNA, cutting of DNA at specific locations, amplification of gene of interest using PCR, insertion of recombinant DNA into the host cell organism, obtaining the foreign gene product, downstream processing, then summary. Please like, share, and subscribe. Thank you so much for watching. Thank you so much for watching.